So this is the column. The white thing that you see in between is the column where the DNA gets trapped. Okay, you can see it and pass it on. So where the DNA gets trapped, we wash it with the buffers, extra buffers, but the DNA remains trapped still, and the other debris like proteins and other organelles and all that stuff gets washed away. We add elution buffer on it, right? And after that, the DNA gets separated from the silica gel with the help of elution buffer, and it gets poured down into the collection tube. So desired DNA is present in collection tube after adding elution buffer. So one is this silica based, and I talked about magnetic bead extraction. The instrument that you see behind, that is in the blue color, it is automated DNA extractor, right? Human intervention is not there at all. Only human work is to put the sample, put the kit, give the instrument a protocol. Then you do not touch anything. Yeah, sir, if you want to see. So everything is being done by a robotic arm. So any pipetting error is not there.
DNA
any type of
tube.
contamination is not there. Any forgetting of reagents or putting anything is not there. High quality, good yield of DNA just gets extracted within 45 minutes. Okay. Everything is controlled by robotic arm. All the conditions are by computerized. Right? And generally in that, magnetic bead extraction technique is used. Right? So extraction, this two are the, Again, as we see nano drop over there, in that system only one sample at a time. One advanced system is also there. 16 samples can be done at a time. This is citation 5. The whole plate is there of 16 samples. Just put it within 10 seconds, it will read the quantity of the DNA and it will be giving to you. Right? Just 2 blanks, and if, for example, I have 100 samples to be quantified for the DNA, I'll just take 2 blanks, initial blank of the system, like elution buffer as a blank, and then I'll put samples, 16 samples, DNA quantified, remove them, again put 16 samples, remove them, continuously hundreds and hundreds of samples could be quantified just by putting 2 blanks. Whereas we have to keep on changing the blanks in the unit protocol. But this doesn't require it. So it's quite one of the advanced version of the nanodrop. Right? Now after extraction and the processing, we go for PCR. PCR is polymerase chain reaction, but it is a technique. Instrument used for that technique is called thermal cycler. As the name suggests, temperature is continuously being changed into a particular cycles. Right? So this is a thermal cycler. Okay. This is the father version. The more advanced version over there is that one, the one blue with the handle up. Yes. It has an advanced technology which is touch screen and all that stuff into it, but performs the same function. Right? This also works as a PCR working reaction area. That also works as a PCR working area. Main basic thing is it keeps on changing the temperatures according to our requirement. That is denaturation temperature at 94 degrees Celsius. Annealing might be at 50 to 50, 60 degrees Celsius, or an elongation of the process that is at 72 degrees Celsius. Right? So it just keeps on cycling that particular conditions to increase the quantity or amplify the DNA between the particular primaries. The instrument just beside it, that white box type of a thing, is a cousin of PCR. Right? The only difference between that PCR and this PCR is it's like a normal PCR, this is real-time PCR. The only thing that added in technology is a fluorescence detection system. With the help of that advancement, you can increase the versatility in the PCR reaction. Right? How? You can identify with the help of fluorescence each and every cycle, how much quantity of DNA is being amplified. Right? So at first cycle, the fluorescence will be less. As intercollecting dye is there, like ETBR, there is one cyber green dye is also there, which is used over here. It is also intercollecting dye. Once first cycle is done, PCR, the DNA quantity has been from 1 to 2, 2 to 4. So the fluorescence will steadily increase. It will detect the fluorescence and it will tell you, okay, in 20th cycle, how much quantity of DNA was there. So in real time, it will tell you how much DNA is getting amplified cycle by cycle. Right? Now, major application of for in forensics for this instrument is for human quantification of DNA. Why we are getting specific for human quantification of DNA is as we all know, crime scene is there and it's highly contaminated, right? And sample size, yeah, obviously. Let's keep a challenging sample like a BD sample, right? So if I get a BD from a crime scene, I take a BD, okay, I extract DNA from that particular BD, right? My colleague is right now working on that process only to standardize that process. So we extract DNA from that BD. Now BD itself is a leaf, so plant DNA will be there. Obviously microorganisms will be there, microbe DNA will be there, as well as the person who has smoked it, right? So human DNA will be there, human epithelial DNA, right? So 3 types of DNA there. But when we quantify over here, we get all the 3 DNA being quantified together, and we say, "Oh, what a quantity of DNA I have achieved." But how much is particularly of human? We don't know that, right? So this instrument, as PCR uses primer, we use human specific primers. So it will only amplify particular human DNA only, right? So cycle by cycle, as the human DNA increases, so we can quantify actual human DNA which could be amplified. Now, why we use it in forensic laboratories is human DNA is important for us. Why it is important? We need to identify that particular human, right? So this is the instrument which does that process. The process that we call DNA fingerprinting, STR profiling, okay, either maternity-paternity testing. It is all done on the basis of short tandem repeat profiling. Okay, our chromosomes are having a particular repeating sets in particular locus or areas. Companies have identified it and they have standardized the technique. One of them is global filer. It works on 24 markers, that is 24 regions in our chromosomes, 23 chromosomes. They identify 24 research regions which has STRs, that are short tandem repeats, ATGC repeats are there, particular types. So, for example, in you it is 5 repeats, for me it's 6 repeats. How it has been inherited from my father and mother, it will be like that, right? So we are getting individualized through that process. There are 24 markers and there are many probabilities that
it is the number of markers on which we decide that the particular DNA belongs to who.
But there are 24 markers. So probability of getting your marker and my marker at the same STR is very big. So unless and until it's an identical twin, okay, which also has its own method of identifying both of them individual. But in STR profiling, unless and until it is not an identical twin, you can identify each and every individual. Yeah. So this instrument helps us in doing that. This is 3500 genetic analyzer, okay, and it helps in identification of the human by the help of specific allelic STRs. Other than that, the application of this instrument is to identify single base pair sequences of the DNA. For example, I had done my PCR, I developed a particular amplicon. I want to know what is the sequence of that amplicon, right? Then I'll go for this instrument again. With the help of a particular reaction that has been developed by Sanger, that is Sanger sequencing reaction. Please go on YouTube, more visually you will see it and you will understand it more properly. So just go on YouTube, search Sanger sequencing, and the idea will be very clear how the Sanger sequencing occurs and how this instrument is being used. In that, base pair wise base pair, you can identify what sequence of ATGC is there in our DNA. So this instrument does 2 major workouts, that is identification of humans through STR profiling and getting the sequencing of the DNA. Yeah, we are into the process of procuring next generation sequencer that helps us to sequence whole genome of any particular organism. So that is still in the procurement phase. So might be when we visit next time, we might be having it in our lab. So till now, this is the cutting edge technology that we have it in our lab. It's this. Any questions regarding this, please feel free to ask. प्लीज कुछ भी क्वेश्चन हो पूछो, डाउट से मत निकलो इधर से.
So what about BNTR?
VNTR, variable number tandem repeats. That could be identified through gel, whereas where the primer gets attached in VNTR, it's the single primer game. So at variable places, the primer gets attached, and it might be, generally VNTR is used to identify subspecies in microbiology, where, for example, sanguinus is there. Sanguinus is a species of bacteria that is found in our mouth, but there are 12 subspecies of it. So, but how will you identify? Almost sequences are the same. With the help of VNTR, we could identify 12 subsets of that particular species. How? A particular small primer is being used. Okay, 10 base pair primer is used, only one primer, and it is being run into 40 cycles or something like that. A standardized protocol is there for that, which gets randomly attached. That particular primer gets randomly attached, and the random products are being formed. Now, as the sequence might be varying from one base pair also, that primer might not attach over there. So what will happen is that product will not form. So when we open into the gel, when we load the gel of that particular reaction, we will find all the various types of products that has been formed by that particular primer. But where that one band is absent in that particular, both the species are same, but one band is absent because of few base pairs are not changed, or substituted, added, or deleted. So because of that, one base pair is one fragment is absent. So upon that, you can individualize subspecies also, where VNTR comes into role.
Yes, sir. Northern blotting, southern blotting, ये क्या होता है?
That is generally used for protein techniques. Isolation of proteins, or as well as DNA from the gel, also is being used by that. But that right now we do not perform it. Right?
Old techniques है. Older techniques है क्या?
मतलब if there is a gel, a particular DNA, you want extract out from the gel or particular protein from doing 2D SDS electrophoresis, you get a particular protein, you want to sequence that protein also. So it's not easily identified from the mixture of the proteins. So once you separate the proteins and do northern blotting or southern blotting, and then just take the protein out of it and then use it for sequencing or LCM, SMS, whatever.
So thank you, sir.
Oh, my pleasure.
Okay. Thank you.